Hepatitis B. Scenario. In our scenario, Nicole visits the clinic to get her blood taken. Nurse Joy enters the room to take her blood. Everything goes smoothly until the end. Nurse Joy doesn't properly dispose of the used needle, which results in the contaminated needle entering Joy's skin. 1 month later, Joy starts to show symptoms of hepatitis B. The first symptom is jaundice, which is the yellowing of the skin. The second symptom is pale stools. Joy wonders what's causing these symptoms.
Risk factors include sharing needles or razors with an infected person, being born to a mother who is infected with hepatitis B, and being exposed to infected blood in the workplace. Joy now recalls her needle prick incident that occurred a month ago at the hospital. She has been infected with the hepatitis B virus from a contaminated needle she didn't discard properly.
Normal physiology. The liver has 4 lobes, the right, left, caudate, and quadrate lobes. The liver is a target organ in hepatitis B. Normally, the liver has a lot of key functions. It produces plasma proteins, specifically ones that play a role in the blood clotting process. It removes body wastes and other foreign substances from the blood, as well as stores vitamins and minerals. An example of how it does this is by using the bile pigment bilirubin. Bilirubin, a bile pigment, has importance in the liver's ability to function normally. When there are dead red blood cells in the bloodstream, macrophages will break down these red blood cells. Bilirubin is a byproduct of hemoglobin when the red blood cell is broken down. The protein albumin will transport the bilirubin from the bloodstream to the liver. It is stored in the bile, which is produced by the liver, and helps the liver digest and absorb fats. It gives the stool its brown color. This is why it is known as a bile pigment. Without a properly functioning liver, we would not have properly filtered blood. Toxins would continue to circulate in the bloodstream, and we would be unable to normally digest and absorb fats.
When an individual is infected with hepatitis B, the liver is constantly under attack by hepatitis B virus. The virus binds onto receptors found on the hepatocyte's cell membrane. The hepatocytes produces more hepatitis B virus, which can spread to surrounding hepatocytes. This virus is causing hepatocyte damage, which triggers the inflammatory response to tissue damage. The hepatocyte's response to inflammation is production of scars. This is known as fibrosis. If fibrosis persists, the scars will elongate and become permanent. This leads to cirrhosis, which is permanent liver damage. The damaged cells are now unable to filter toxins, waste from the blood. These cells are also unable to produce bile, as well as the process of the bile pigment, bilirubin. This pigment now accumulates in the bloodstream instead of being removed with the bile. This causes the symptoms associated with hepatitis B.
In our scenario, Joy is producing pale stools because her hepatocytes have become damaged and are unable to produce adequate amounts of bile and process bilirubin. The lack of bilirubin being excreted out of the body is what causes the pale color of the stools. The second symptom Joy experiences is jaundice. This is because the bilirubin, normally a yellow color, is accumulating in the bloodstream instead of being put into the bile and excreted out of the body. This is what gives the skin that yellow pigmentation.
Key concepts. Cell membrane. The cell membrane plays a key role in allowing hepatitis B virus infection. The cell membrane is where the surface antigen of the hepatitis B virus bind onto to release the core particle of the virus in the hepatocytes or liver cells. Homeostasis. Our body's immune system works hard in warding off the hepatitis B virus. The immune response to the infected or damaged hepatocytes is inflammation, which can become cirrhosis over time or permanent scarring of the liver tissue. The scarring of the liver tissue prevents the blood to flow normally that could result to cell death due to decreased delivery of nutrients and decreased production of plasma proteins for blood clotting. Another way hepatitis B disrupts our homeostasis is hepatitis B virus infection prevents the liver from producing the needed clotting factors that work on hemostasis. Protein. The surface antigen that encapsulates the hepatitis B virus bind onto the cell membrane of the host cell, allowing the release of the core particle of the virus. Without this protein, the virus cannot replicate.
Let's go back to the original scenario. Joy's immune system, specifically her T cells, would destroy the infected hepatocytes and aid in ending the infection. But if her immune system isn't effective at removing these damaged cells, the infection will persist. Her treatment option would then be antiviral medication. They function to slow the virus's ability to replicate and infect other hepatocytes.